All right, we'll get started here. Chemistry, you all took the chem prereq, so what I usually do is I just cover the biochemistry part, the part that's a little more relevant to A and P. And so some of this should be reviewed for you though, but I will cover what I think is relevant for 430, 431. And I'll start with what we eat: carbs, fat, and protein. I'll start with the study of organic compounds. There's a picture of each of those molecules. I'll start with the carbohydrates. Carbohydrates so I usually, because of that molecular formula, I usually just abbreviate carbohydrates, CHO sometimes. Anyways, the monomers of carbohydrates are the simple sugars or monosaccharides. And the monosaccharides include, let's look at the three on the left: glucose, fructose, galactose. Okay, we'll do those three. I'll talk about the other two, these pentose sugars, when I talk about DNA. I'm going to turn my mic down a little bit. Glucose, fructose, galactose. And I'm more concerned, you know, the names, not necessarily the molecular structure. And particular no glucose, that's the circulation form of sugar, known as blood sugar. Okay. Well, we can link up the monosaccharides. In a dehydration synthesis reaction, water is given off to link the two to form disaccharides, and this is reversible. Hydrolysis is the insertion of water to separate the disaccharide into two monomers. But okay, let's learn the different disaccharides that are commonly found around us: sucrose, maltose, lactose. Sucrose, you don't know what it is composed of. A glucose linked to a fructose. I'll just put glu plus fruit to remind you. Well, that's basically table sugar. And then you have maltose, you know, malt sugar. Barley, and its two monomers that are linked together are a glucose plus a glucose. So glu plus glu. Lactose, that's the sugar in milk. That's a galactose plus a glucose. So I'll put gal plus glu. So those are the three disaccharides you're responsible for. Well, the way the body stores it, you consume sugars, and your body stores them. We call them polysaccharides. Really, it's starch in plants, glycogen in humans. So let's put that down. It's a storage form of carb. So those simple sugars circulate. Muscle, liver, they take those, they link them together, they store them in these long chains, and no glycogen. Storage form. Of carb, CHO, in animals. It's stored in skeletal muscle and liver. Let's say you increase your activity level, you go work out, and glucagon, a hormone, will kind of like target this. It'll break those bonds and it'll liberate the simple sugars from that long chain of sugars into your bloodstream so your muscles can use it for energy. But this is how you store it. You either store it or burn it, right? Eat a large meal, we call that the insulin trigger. You store all that, and if it's carb you ate, then it's stored as glycogen. In plants, it's starch. So we eat the starch, it's digested, we store it in that form. So I'll just put storage form of carb in plants. Now let's move on. Lipids, which taste very good. In terms of a chemistry lecture, these are the forms we got to know. Triglycerides, actually cross that one out. Eicosanoids, I'm not going to test you on. Guys, save it for later. For now, don't worry about it. Phospholipids, glycolipids, yes, and steroids, yes. We will talk about that today. Now, they also are composed of, fats are composed also of carbon, hydrogen, oxygen, but the ratio is different. So let me just erase that. Basically less oxygen. Okay, so let's talk about triglycerides. This is the fat that makes you fat. It's the storage form of fat in animals, okay? Tri implies what number? 3. What we see here is this glycerol backbone. Let me get my pointer. This glycerol backbone, this 3-carbon glycerol backbone, and there's 3 fatty acid chains. These are hydrocarbons attached to it. So that's kind of how we describe its structure. 3-carbon glycerol backbone. Quote unquote, you know. But then it's got 3 fatty acids at each of those 3 carbon positions. What's that?
What does A mean? A3 carbon?
Oh, just mean that it is 3-carbon glycerol backbone. So with the glycerol backbone with the 3 fatty acids. So when they're attached, now this is how you store fat in your body. So if you want to burn the fat for fuel, you have to liberate the fatty acid, cleave it off of there. Okay. So to build the thing, you attach the fatty acid to the carbon position, giving off a water molecule. So you attach all 3. This is how your body stores it. And I think we know where the fat is on our body, right? It's under our skin. It marbles in our muscles. Fat is everywhere. Fat stored. Subcutaneously. Marbles muscle. can see that in the steak you eat, how it marbles. It's first stored in the muscle, and once the marbling is, you can't hold any more in your muscle, then you start to deposit it under your skin. Now the different forms of fat we consume, when we say, you kind of read it on your nutrition labels, saturated fat, let's mention that. Saturated fat. What they're referring to are those hydrocarbon chains that are fully saturated with hydrogens. There's no double bonds. That's basically what we're saying. No double bonds in that chain. So I'll say fatty acid, quote unquote saturated, well, with hydrogen atoms they show like a cube of butter, right? Because of the structure, no double bonds, the chain is relatively straight. Makes for a straight chain. Which means you can pack them tightly together if they're straight, and just kind of pack tightly together so that at room temperature they're solid, right? Like butter. Of course, you can melt the butter, but at room temperature it's solid. So foods that are high in saturated fats tend to be solid at room temp. So fats that contain unsaturated fatty acids have double bonds in there, and that puts a kink in the chain, and that allows more space to be in between the fat molecules, like right here. And then so at room temperature, like olive oils, they're liquid. Unsaturated fats, a couple varieties. They can be monounsaturated or polyunsaturated. Poly means more than one double bond. Mono means one double bond in the chain, like this one here. Monounsaturated. But let me sort of put that. Polyunsaturated. I'll put more than one dB, double bond. Monounsaturated. One dB. So basically, liquid at room temperature. Saturated fats are less heart healthy. They raise cholesterol levels more. They call the unsaturated fats the healthy fats, but I wouldn't use the term healthy. They just don't raise cholesterol quite as much. Okay, those are kind of fats that we eat, butter and oil. Now, in terms of biology, some lipids that are important to us are the phospholipids. The structure is shown here. So you can look at the picture for the structure, but what you see if you look at the left there, there's also a 3-carbon glycerol structure right there. And you have the 2 fatty acids at these 2 positions. In this position here, you have a phosphate group, so that's the difference. So you have glycerol. You have two fatty acid chains. Those fatty acid chains are considered the nonpolar hydrophobic tails, shown here. You should probably note that. Nonpolar hydrophobic tails. And then the other carbon position is the polar hydrophilic phosphate group. So the phosphate group it's polar or hydrophilic, water loving. It can face water. Okay. And so we like to introduce phospholipids early in the course because they form a lipid bilayer. And so that's usually how they're illustrated. They usually just draw the polarized head group and draw them next to each other. They're usually a double bond in one of the fatty acid chains. That'd be one layer. Then there's another layer that faces it. So this lipid bilayer is basically cell membrane or plasma membrane. And it's important for cells to be a membrane barrier where the hydrophobic tails face each other. Well, they call that the hydrophobic zone. Polar molecules can't pass by there. So things like ions and proteins, if they are to get in and out of the cell, they need a protein transporter. Okay, so that zone is important for that reason. There are different types of phospholipids. The most common one that form this lipid bilayer in the cell membrane is called phosphatidylcholines. Let me write that on the board. So maybe as a type of phospholipid, phosphatidylcholine.
I mean, you refer here then to the head cell of those, they are called.
Oh no, I'm referring to the entire molecule. So what I'm saying is phospholipids is a class of lipids. There are many different kinds. So I've given you one kind to know. Phosphatidylcholine, which is typically the phospholipid that forms the cell membranes in your cells. Okay? So there's a hydrophobic zone, I already went over that. So notice that the polar heads will face the water side, okay? Whether it's outside the cell or inside the cell. And it's the hydrophobic tails that are turned in on each other, forming that hydrophobic zone. I say you didn't have to know eicosanoids. Don't worry about it. I'm just going to fly through those slides and not even give it a thought. But I will spend some time discussing the different steroid hormones. Steroids are lipids, but they have a much very different structure than triglycerides or phospholipids. They have this 4-ring hydrocarbon ring structure. That's kind of what we call it. It's a steroid. This is actually cholesterol. But most steroids are derived from cholesterol. Steroid hormones are derived from cholesterol. Now, cholesterol is produced by the liver, from liver. And we always hear cholesterol, oh, it's bad for your heart. It's always a health issue. That's what you need cholesterol, okay? So what happens is we have to remember that lipids are hydrophobic. They're not very soluble in water, and your blood, plasma, is a very watery medium. So what we need to know is we need to find a way to transport lipids like cholesterol in the body in a way where it can be transported in the blood. And that's how you always hear about LDL, HDL. Low density lipoprotein, high density lipoprotein. It's a way to transport triglycerides and cholesterol in the bloodstream. And what they are is just a bunch of phospholipids in a circle. I'm going to put the phosphate heads facing out and the tails face in. And in the core, in the lipid core, you would have things like these steroids, these little ring structures. So I'll draw like a little ring structure in there. I'll draw some triglycerides. Okay, the point is that's how the body compensates for lipids being not water soluble, but you can transport it around. And LDL is associated with heart disease. HDL is actually preventative, prevents heart disease. So they call this the good and bad cholesterol. I just wanted you to know where that came from. I think what you need to be concerned about as well is that steroids are derived from cholesterol, and they're called the corticosteroids. And you'll learn those a lot in endocrine in the first unit of 431 next time. Okay, so steroids are derived from cholesterol. They're called corticosteroids. We'll learn about those next time. There's also the sex hormones as another category of steroids. When you learn about the corticosteroids, you'll learn about the adrenal cortex that secretes them. Just kind of put some physiology on this. The adrenal cortex is right above your kidneys. Okay, and those corticosteroids really help you deal with stress. The sex hormones, estrogen and progesterone, shown there, on bottom, they put a little crown there because that's the king androgen hormone for males, but there's estrogen. And you can see the structure is very similar to cholesterol. Also have sex hormones. So no estrogen. So female, gender, and testosterone. So a couple of sex hormones. Or male, okay. All derived from cholesterol. Steroids are lipids. That's the thing you gotta know. And then you have proteins. And proteins, the basic structure is the amino acid. The amino acids, your body can synthesize some of them. Cells can make them. They're called the non-essential ones because your body can synthesize them. But there are some essential ones that you can only get from your diet. So for the first thing I said, these are the monomers of proteins. The non-essential amino acids, your body's cells can synthesize them. Non-essential non-essential. Body can synthesize. The essential ones must be consumed by diet. Essential amino acids. Consumed by your diet. So a protein that's a complete amino acid, it's got all of them, essential, non-essential. I want you to know the basic structure of an amino acid. There's like 20 different ones, and if this were a biochemistry class, we'd spend a whole unit talking about the chemical properties of the different 20 amino acids. Just know the basic structure of all of them. You know, essential carbon, surrounded by 4 positions, right? And okay, you have I'll just draw it like this. That's a carboxylic acid. At one position. You have an amine group at another position. A hydrogen at a third position. At that fourth position where they put a generic R, that's actually the functional group. It gives the amino acid its characteristics so the protein can fold in a certain way. And they give some examples of amino acids. I said there's 20 of them. They show you 5 here. Well, they show you 4. The first one's a generalized structure. Then glycine, aspartic acid, lysine, cysteine. And we're not going to learn really the names. Just understand that you link these together to form proteins. If you just link 2 together, it's a dipeptide. Di is 2. Write that down. Amino acids linked by a peptide bond. Know that bond. I want you to know peptide bond. Right there. That's the bond that holds proteins together. Now, I have a picture of the 20 amino acids here, and again, you don't have to know the names, but understand the building blocks of all proteins come from linking these together. And the diversity of proteins, I have a list of them here. I mean, we have to know, okay? And so the diversity of proteins in biology is a lot. Let me go through some today. For example, link together proteins. To be a structural protein, you have to be well, let me just give you an example. A collagen is an example of a structural protein you see in your body cells or tissues. So let me list examples of proteins in biology. The kind of proteins you're familiar with is, I guess, what you eat. Chicken, fish, steak, right? Plant proteins too. Soy and tofu, but what your body does with it, you eat it, you consume it, you digest it, and your body makes it into other things that are useful for your cells and your body's tissues, like collagen, structural proteins. There are others. When we get to tissues, we'll learn different fiber types. But just to list one from now as a category. Another category are enzymes, enzyme proteins. Enzymes, if it ends in the suffix quote unquote "ase," peptidase, something ACE. Well, what it does is, as a general category, enzymes, for example, it's breaking this down or putting it together. So you can see that the substrates, these shapes, they fit in the enzyme. And what it does is it speeds up the chemical reaction rate. So let me put that as a general thing of what we want you to know about enzymes, what they do. Speeds up chemical reactions. By lowering the activation energy. I think there's a slide showing you a graph of activation energy being lowered, so that you can see without the enzyme, it takes more energy required to get the substrates to form the product. You know, it's a bigger hill for the reactants to get over, so that would be a slower chemical reaction. If you can lower the activation energy, then it will be much faster. That's the basic idea. Other things that proteins can do, they're transporters. Transport proteins. The main one you'll learn about is like hemoglobin, as an example. Transports O2, CO2 in the blood. Or muscle. Muscle is protein that we all know about. Contractile proteins. Muscle. And what you see pictured there are what's called the actin and the myosin filaments. Those are muscle proteins. Actin filaments, myosin filaments. So these are fibers, filaments. That's kind of what the word implies, and we'll learn the physiology later. Just as an example, sometimes we call these the thick and thin filaments. The actin's a little bit thinner. Look at it under a microscope. Thin, thick. So that's an example of contractile protein. In terms of cell physiology, they can be communication. Pretend this is generic cell, and the cell membrane is made out of lipid, as we just said, phospholipid. And you have hormones that try to communicate with cells, and whether or not some of them are not lipid soluble because they're proteins and they can't get through the lipid bilayer to turn the cell on. So they kind of have to act through these go-betweens, like these receptors. And they call it here, a chemical messenger that the hormone can act through. So let's say this is the hormone that can't get through the cell membrane. It would just bounce off. Okay, but if it has a chemical messenger that it can act through, it doesn't ever get into the cell, but is acting through other things to shut out to start off, some kind of intracellular chain of, for example, G proteins, things like that. So my example is a hormone acting through a receptor. So the communication protein is the blue structure I drew. Hormone acting through a receptor on the cell surface. Because it's not lipid soluble, it can't get in. That's a good example. Defensive proteins. Okay, what's shown there are antibodies or immunoglobulins. Those are proteins. Defensive proteins, example, immunoglobulins. Antibodies, as they're commonly called. Your body is always building an immunity, a defense against anything that is non-self, foreign to the body. It has the ability to recognize it. So let's say this a structure is foreign to the body. Your immune system has these floating around in the bloodstream. They're made by the B cells. Secrete antibodies. They're different classes. We'll just say antibodies is a generic term for now, and they're specific for a bug that's considered non-self. I'll say attack foreign invaders. We'll just leave it at that. You'll have a whole chapter on immunology later in your career when you get to 431. But we will spend a lot of time in the next chapter talking about these kinds of proteins that do transport work. So proteins as transporters for polar hydrophilic ions or molecules. That's basically what you see illustrated in transport work. So let me draw one. Let's pretend this is my protein transporter, and it's embedded in a lipid bilayer as shown in the picture. I'll just draw like this lipid bilayer. And let's say you're a sodium ion that carries a full positive charge, and a polar molecule or an ion like sodium that has a full positive charge, it can't get past the hydrophobic zone. Okay, so if it is to be allowed entry into the cell due to its concentration gradient, I guess what they don't tell you is this transporter that's a protein, it's usually lined with water. this pore. Pore lined with water. So things like sodium, which are water soluble, can pass through. They can't get past the hydrophobic zone. They can get through that. Okay, so that'll allow sodium to be transported across the membrane. Just to give you an example of why you need proteins as transporters for cells. Because there's a lot of things in biology that are hydrophilic and polar.
This is basically the idea how the sodium acid pump works.
Yeah, that's one example of a pump that I'll talk about in chapter 3, yes. That is correct. And this one they have in the picture, this is an example of a channel, it's a pore. This one is more like the one you're referring to, where if you're moving it one way or the other, it may require energy. Here, that's what they're showing you there to do the transport work. But I'll be more detailed about that in another lecture. So today the concept is just get sodium across the membrane using a protein. If it's not there, you can't do it. So the structure of proteins, well, we teach the level of protein structure. And we start with the most basic thing that you can understand. Just link the amino acids together in a particular sequence. That's what cells do. When they transcribe the instructions for the gene that's turned on, use your RNA transcript, and you link the amino acids together to make the protein. The sequence is important, and that's the primary structure. Protein structure. So the primary level shown here, strictly the amino acid sequence. If a cell makes a mistake, there's a mutation or something, and that sequence isn't correct, the protein won't fold correctly and it won't do anything. It'll be non-functional. So that's why that's important to know. The amino acid sequence must be correct. Now, if the sequence is correct, it'll start to fold into these generic shapes, so the protein can have some function. So this is the secondary structure. When the amino acids are all linked together, what you have is a polypeptide chain. That's usually what they call it. And the secondary level is that this chain will start to fold. Fold takes shape. In those shapes that we want you to know, the alpha helice or beta pleated sheet are the shapes I want you to know. Alpha helix, beta sheet. I said pleated sheet because it looks kind of pleated. It's not a straight sheet. Okay. Anyways, what's holding these together are these little dotted lines, which are weak bonds called hydrogen bonds. Held together by hydrogen bonds. All right, so the tertiary structure is these alpha helices, beta pleated sheets. As the polypeptide folds more and more and more and more, you may have many of these in a row or in series. And that's kind of the tertiary structure. It's one polypeptide chain with many of these helices and sheets in a row. Tertiary. It's one polypeptide chain. Still, that's key to know. Tertiary structure is still just one polypeptide chain, but it consists of many alpha helices and beta sheets in a row. Alpha helices and beta sheets in a row. Maybe have a helix, and then maybe a sheet, followed by another sheet, followed by another helix. But the point is, from beginning to end, it's one sheet with many things in series. Still one polypeptide chain, and that's what they're showing you there. They even give you some examples. So the quaternary structure is where you take a few of those tertiary structures and you link them together. And that's called quaternary. The quaternary structure is more than one polypeptide chain. Linked together by things called, like disulfide bonds. So disulfide bond is a very strong bond. Let me just go SS for disulfide. Maybe it links that polypeptide chain to another one, right? I'm not trying to be real seriously organized about my polypeptide chains. The concept is more than one polypeptide chain linked together by a strong bond, like a disulfide bond. That's the overall concept of the structure. And like for example, if I were to show you these images, having taught the basic idea, like in your notes, could you just guess correctly which one A is, B is, and C is? Take a stab at A. What do you think that they're trying to show you there? Which of those choices is it? That looks like an alpha helix. So what's the level? B. Now I look at this and I see several alpha helices in a row, but it looks like it's just one chain because they only use one color. So what level did we define that to be? Tertiary. Now this one here, if you look at your clues, I see 4 different colors. These look like long chains each. I know this is hemoglobin. I happen to know this is coronary because you have more than one polypeptide chain linked together. Okay. All right, so more review on bonding is next. And so I'll be real quick about this. I know you guys learned about this in Chem 305 or 309 or 400 or whatever you took. Let's remind ourselves that ionic bonds involve the transfer of electrons. That's what's shown here between sodium and chloride. So bonding review. sodium. Mhm. Okay, ionic bonding involves the transfer of electrons. And electron. It results in, I guess, ionized molecules. Results. Forms ions. Easy for sodium to give up its outermost electron, give it to chloride. But after the exchange, you form the ions. Sodium will have one full positive charge, and then chloride will have one full negative charge, and positive, negative, attract, and that's the bond between the positive and the negative. Okay, so that bond is when the positive is attracted to the negative, and that's more of the weak. It's a weaker bond, that positive and negative attracted to each other. In other words, it would take less energy to break it. It's not a very strong bond, the bond between positive and negative. Yeah, so there's the positive and negative forming sodium chloride. Covalent bonds are a bit stronger. It involves the sharing of an electron pair. The atoms share an electron pair. So on this slide, let me review what I have on it. It involves equal sharing of electrons. It creates nonpolar covalent bonds. These bonds are very inert and hydrophobic. So there are a lot of words here. But the key is when you share the electrons, the sharing can be equal or unequal. So what that really means is these electrons are spinning around the nucleus. But when they share the electron pair, if the spinning of the electrons is pretty much equal in time around both, the molecule as a whole pretty much is nonpolar. So let me write that down. If it's an equal sharing, I guess that's the key where I put quotes around it. It's just spinning around these atoms equal time. Okay. If equal sharing occurs, overall, the molecule is, we say, inert. It's not polar, it's inert, it's non-polar. Inert is a word that's used, non-polar. I'm just trying to show you all the words that are used. And they give a couple examples here. Nitrogen, oxygen's a good one. Okay. Good old O2. The molecule of life that our blood transports is relatively inert. It can be transported by the hemoglobin. Okay. But in biology, it's important that some things are not like that. Okay. CO2 is another example of a nonpolar molecule. O2, CO2, your blood gases, they are nonpolar molecules. Water, on the other hand, is an example of a molecule that has a slight charge, a slight positive, slight negative charge, because the covalent bonds between the H2O, is covalent bonds that are unequal sharing. So that's a good example. Water, H2O. An example of unequal sharing. So that means the electrons are spinning a little more time around the oxygen and not the hydrogens. it has to do with the properties of the nucleus of oxygen. It kind of pulls the electrons closer to it as it spins around both. But that gives the oxygen a partial negative charge because the electrons spend a little more time spinning around it. And it makes the hydrogens a little bit partial positive. That's why they use this little delta is like a partial negative charge, delta partial positive charge. It's not a full charge like sodium or chloride. It's full negative, full positive, full negative. So that's why they kind of use this symbolism. Delta positive, delta negative, partial charge. And what that does is it creates liquid water. Liquid water is the hydrogen bonding. For example, this partial positive charge on this water molecule would be weakly attracted to the partial negative charge of a neighboring water molecule, and you symbolize the hydrogen bond with a dotted line like they do in the picture. Okay, so no hydrogen bonding. Hydrogen bonding. I'll describe it as weak bonding. Between a partial negative, partial positive charge. Partial negative, partial positive charges. And water is a good example. Here's another slide showing you the hydrogen bonding of water molecules. So what I say on the slide is water is a hydrogen bonding environment. Any molecule that is polarized can interact with water. It is hydrophilic and soluble in water. Non-polarized molecules like fats, for example, tend to be hydrophobic and insoluble in water. Okay. All right, so that gets to the subject of polarity. I think I already defined these terms. Ionic bond and covalent bonds. Polar covalent, that's the unequal sharing thing. Non-polar covalent, that's the equal sharing thing. All right, so that's a nice little table summarizing that. Let's see here. And I put some other examples on the next slide, asking you if it's polar or non-polar. Molecules tend to be polar if they have these elements. Okay, and all of these are examples of molecules in biology that are polar, and a lot of the lipids that we talked about today tend to be non-polar. Okay, water hating. And I wanted to give you one slide of the 20 amino acids that I showed you before, and I won't ask you this on the test, but just to get the concept across, since it's color-coded, it's easy to ask this question to the class. Which group is the most hydrophobic? And it's good to know as a student of biology, molecules that tend to have more hydrocarbons or ring structures tend to be a little more hydrophobic. So the answer is orange. That's important for cells. If you're going to make proteins, sometimes you want to make proteins that have a lot in that orange group so that proteins can kind of span the width of the cell membrane. Because remember, there's a hydrophobic zone. So if you have hydrophobic amino acids in that stretch, it makes it useful to be inside there. And so then I asked this question here. You haven't had time to process, but I'll ask the question anyways. Which part of the phospholipid is more hydrophilic? Do you think it's the phosphate group or this tail end? Top or bottom? if you had to guess, is the top, and that's the water-loving side. Okay. Looking at the time there, I have some more slides. I usually give examples in physiology. I've decided based on looking at the time, make a note to yourself. All the slides after here, you're not responsible for. Okay, I'll make sure it's not on the test, all right, so don't worry about it. It's time for a break. But when we come back, I would like to lecture on mitosis, okay? So take about a 15-minute break. I'll see you around 9 o'clock.